Assalamu alaikum, everyone. Today, inshallah, we're going to start learning about the taxonomy of bacteria. So, we already discussed in the previous lecture the two major domains of prokaryotes. We have archaea and bacteria. Both they constitute prokaryotes. Okay. And bacteria also subdivided into subdomains or subgroups that are diverse in their modes of nutrition, okay, as we saw in the previous lecture. So the first group of bacteria is called the proteobacteria. So remember, we have 5 groups of bacteria, or 5 groups of the domain bacteria. We have proteobacteria, chlamydias, spirochetes, cyanobacteria, gram-positive bacteria. So you're going to learn in details about members of these groups and where they are found and how they are implicated in health and disease, inshallah, in a course, second-year course called microbiology. In this course, I need you to know that there are 5 groups of domain bacteria, and these groups, they are classified based on their based on genetic and phenotypic characteristics. So proteobacteria is divided into 5 subgroups: alpha, beta, gamma, delta, and epsilon. And here we can see 5 different representative, let's say, organisms of these 5 groups. Proteobacteria includes gram-negative bacteria that are considered photoautotrophs, chemoautotrophs, and heterotrophs. Some members of this group can go in the absence of oxygen, and some in the presence of oxygen. Okay. We have another group of the domain bacteria called cyanobacteria. It's a gram-negative bacteria, and this type of bacteria, they can carry out photosynthesis, and they are considered photoautotrophs. They can generate oxygen. Okay. Typically, cyanobacteria they are found in fresh water and in marine waters. So this is a picture of filaments of a type cyanobacteria called oscillatoria. Okay, and as you can see, from inside, it has these green pigments, just like plants, and these green pigments allow this bacterium to capture the light energy. Then we have gram-positive bacteria, such as actinomycetes, and we have some, and these actinomycetes, they are found typically in soil, and they decompose organic matter. So they can decompose organic matter into simple molecules. So for example, when an organism in the soil dies, these actinomycetes, they can decompose or degrade the organic matter from the dead organism or dead animal into simple molecules or inorganic molecules, okay, that could be used by other live organisms as a source of energy and a source of nutrients. Other examples of gram-positive bacteria, we have Bacillus anthracis, which can cause the anthrax. It's a deadly disease caused by the spores of this type of bacteria. We have Clostridium botulinum, which causes botulism. It's a type of disease. This bacteria can secrete a toxin that can cause disease to humans. And we have other types of disease-causing bacteria, like disease-causing gram-positive bacteria, like Staphylococcus aureus and Staphylococcus and Streptococcus species. Members of the gram-positive bacteria also include the mycoplasma, which are the smallest known cells. And these cells, these bacterial cells, they do not have cell wall like the majority of bacteria. This is an electron microscope image showing the spores or the filaments of a gram-positive bacteria called Streptomyces, which is a member of the colony-forming group called actinomycetes. Archaea, they have characteristics that are shared among bacteria and eukaryotes, but they have their own unique characteristics that are not found in bacteria. If you see in the evolutionary tree, Archaea is between eukarya and bacteria. This table compares between bacteria, archaea, and eukaryotes or eukaryotic cells in terms of many characteristics. So as you can see, some of these characteristics are shared between bacteria and archaea, like the absence of nuclear envelope, the absence of membrane-bound organelles. But some other characteristics are shared between archaea and eukaryotes, like the absence of peptidoglycan cell. Okay. Some archaea, they live in extreme environments, very harsh environments. Normally, other organisms cannot withstand. That's why these archaea that live in extreme environments, they are called extremophiles. Philes means loving. Okay. Some extremophiles, they are called extreme halophils. Halo means salt. And they can live in high salty environments. We have thermophils. They can live in hot environments like hot springs. مثل حمامات الشونة أو حمامات ماء. Methanogens are also examples of an example of archaea. These methanogens, they live in swamps, مستنقعات, and they can produce methane gas as a waste product. So methanogens, some methanogens, they can use CO2 as a final electron acceptor. So they carry out anaerobic respiration. Okay. And plus H2, they can produce methane gas, CH4, okay, plus water. Okay, so this is not a balanced equation here. We have 2, 4. Okay, now it's balanced. Now, methane, by the way, is one of the gases that is implicated in global warming. Okay. So methanogens, they are strict anaerobes. They do not use oxygen as a final electron acceptor in the electron transport chain. They use CO2. Okay. Some they might use acetic acid as an electron as a final electron acceptor. Okay. We understand more about bacteria than archaea. Still, scientists are studying archaea, trying to understand how basically they can withstand living in harsh environments, for example. And scientists are still looking and trying to compare archaea and bacteria and they're trying to basically establish a connection between these two groups of prokaryotes. The last concept in this chapter, we're going to learn about some of the important roles played by prokaryotes in the biosphere. Okay? So, without prokaryotes, life will not exist on Earth, because these prokaryotes that are found around us everywhere, they make our life more easy, and they help us with cleaning the environment. So many prokaryotes in the environment, they carry out what we call chemical recycling. So chemical recycling is basically a process where prokaryotes help recycling chemical elements between living and from non-living components, or from dead animals, for example, in the environment, or non-living components, so that they could be available for living organisms, and living organisms could basically benefit from these chemical elements. Some prokaryotes are chemoheterotrophs, and they can carry out decomposition, or they are considered decomposers. They can break down dead organisms and waste products, releasing the chemical elements from these waste products or dead organisms. And these chemical elements will be available for other organisms, and they could be used as nutrients for other organisms. So we know that most of the energy on Earth comes from the sun. Okay, so the solar energy is captured by plants. So plants, they can carry out, plants and some bacteria like cyanobacteria, they can carry out photosynthesis. Okay, and they can use the sun energy and CO2 from the atmosphere, okay, to synthesize food, okay, like carbohydrates. And then we have some consumers, مستهلكين. Okay, so this is a plant or plants in general. And some consumers like some organisms, they can take the food from plants. Okay. For example, like a grasshopper. Jundub. Okay. Grasshopper or some other insects or other animals, they can feed on plants or the food or the sugars produced by plants. Okay? And then maybe another animal can feed on grasshoppers, like snakes. Snakes, they can. Snakes, for example, they can then eat the grasshopper. So the food that was basically, or the nutrients or the atoms that was once available or produced by plants are now being transferred into snakes. So because the snake eats the grasshopper, okay, and then maybe another. So this is considered a primary consumer of nutrients from or elements from plants. This is a secondary consumer. And then maybe, let's say like an eagle, sagir, okay, will come and eat the snake. So as you can see, the food or the elements, the chemical elements or the nutrients that once were found in plants are now, and the energy that once stored in this food or these carbohydrates are now being consumed by a tertiary consumer. Okay. So now what we are witnessing here, this is how energy and atoms are moving in this direction. Okay. Then at some point, this eagle is dead because all organisms, they will eventually die. And now you have to understand that the dead eagle matter contains atoms that could be released by decomposers like bacteria. So decomposers they will release basically the nutrients or chemical elements, okay, from the dead animals so that they could be used by plants. Okay, these chemical elements and nutrients could be used by plants, okay, to make more food. Of course, water is also needed in this process. Okay. So as you can see here, we have recycling, and an important part or an important component of this chemical recycling process is the presence of decomposer in the environment, because they can release the nutrients or the chemical elements in dead animals or non-living material to be available for other living organisms like plants to benefit from these elements, okay, or from these released nutrients or chemical elements. This is what I was explaining prokaryotes, they can increase the availability of nutrients required for plant growth. Okay. But also prokaryotes, especially in the environment, they can immobilize or decrease the availability of nutrients, so they could use these nutrients for their own purpose. Okay. Now, in the environment, we have what we call ecological interactions. تفاعل بيئي between prokaryotes or different types of prokaryotes, or between prokaryotes and other organisms like large organisms or eukaryotes. So we're going to define the major types of these ecological interactions. We have a term called symbiosis. So symbiosis is basically a relationship in which two species live together. One is a larger host, and then we have a smaller organism called symbionte. So the larger organism is called the host, and the smaller organism is called symbionte. So for example, we have a symbiotic relationship between prokaryotes, which are the symbionte, and larger organisms like humans or other animals, which are called, in this case, the host. Okay. So I told you before that in the human intestine, okay, we have millions of bacteria or millions of prokaryotes, okay, living inside our intestine. These are called prokaryotes and eukaryotes, or microorganisms in general. And this is an example of symbiotic or symbiotic relationship. The human is considered, in this case, a host because it is the larger animal, and these prokaryotes or bacteria, some of these prokaryotes are bacteria, they are considered the symbionte. So the bacteria inside our intestine, for example, they do not cause disease for us. They are not harmful. We provide them with food, and they help us with digesting food, and they might make for us some types of nutrients like vitamins. And some of these prokaryotes, they protect us against harmful bacteria or pathogenic prokaryotes. Okay, so this is a relationship where both the host and the symbionte, they benefit from each other. So this relationship is an example of mutualism. Mutualism means both the symbiotic organisms, they benefit from each other. So just like in this example, the bacteria inside our intestine, they will benefit from their presence inside our intestine because we provide them with food. On the other hand, we benefit from these prokaryotes in many different ways. We have commensalism. This is another type of symbiotic relationship where only one organism is benefits from this relationship, but the other one does not cause any harm. So only it's a relationship between a host and a symbionte, but only one of them benefits from the other, while the other one does not cause any harm. Okay. We have also another type of ecological interaction or relationship, which is the parasitism. Here, one organism, which will be called the parasite, will cause harm to the other organism, but it does not kill it. Okay. So the symbionte will be called parasite, and it will harm the host, but without killing it. Some parasites, they might cause disease, and they are called pathogens. So an ecosystem cannot exist without, or ecosystems, some ecosystems cannot exist without prokaryotes. They completely depends on the presence of prokaryotes. Okay. So an ecosystem, for example, it's an ecosystem is defined as, let's say, all organisms, whether big, large, or microorganisms, and their environment. With which they interact. So basically, an ecosystem consists of the environment and the organisms within this environment and how they interact with each other and with the environment surrounding them, okay, with which they interact. Okay. So there is an example of the ecological community in the hydrothermal vents that depends on the presence of chemoautotrophic bacteria, because this bacteria will provide energy for other organisms in these hydrothermal vents. Okay. So these hydrothermal vents, I don't know what they are called in Arabic, but these are basically openings in the earth crust أو فوهات الحمائية هيك مكتوب. فتحة فوهة حرارية مائية. Okay. So these are, let's say, you have, for example, Earth, and then you have an ocean, let's say, or sea. So this is the ocean or sea, and in the bottom of the oceans, there will be some fissures, okay, or fathat. So in the sea floor, there will be a fissure or fathat, okay, through which there will be like hot water coming out. Okay? So some organisms, they live in these fissures, and they do not see light and they cannot obtain energy. So some bacteria in these fissures or in these hydrothermal vents, they provide other organisms in these hydrothermal vents with energy. So this is an example where prokaryotes help other organisms within the hydrothermal vent ecosystem. And by the way, ecosystem in Arabic, do you know what's the meaning of ecosystem in Arabic? Anyone? نظام بي. Some types of prokaryotes, they can cause illness or diseases to us, so they are considered the human pathogens. But many others, they do not cause harm for us, or they are beneficial to humans. So most bacteria or most prokaryotes, they do not cause harm to humans. On the contrary, some are actually beneficial to us. But at the same time, we have some prokaryotes that might cause disease in humans and in animals or plants. So in our intestines, as I told you before, we have between 500 to 1,000 species of bacteria. And these bacteria are beneficial to us. They can help us digest our food. They can provide us with certain nutrients, and they can also protect us against pathogenic bacteria. Okay? Bacteria or bacterial pathogens, they are known to cause half of all human diseases. And some of these bacterial diseases are caused by bacteria directly or indirectly because they are transmitted by other species. So for example, Lyme disease is caused by a bacteria that is transmitted. So this bacteria that causes Lyme disease is called Borrelia. And this bacteria is found inside ticks. So this is a tick commonly. Okay. So when this tick basically bites our skin, it will inject this bacteria inside our bodies, and this bacteria will cause the Lyme disease for us. Okay. So this is the tick. Some other types of prokaryotes or pathogenic prokaryotes, they might cause disease by producing toxins or by releasing toxins. We have exotoxins or endotoxins. Exotoxins are typically toxins that are produced inside prokaryotes and then they are released. Okay. And endotoxins, these are toxins that are released from the bacteria when they die and their cell wall break down. So for example, we have an example of an exotoxin is the exotoxin secreted by Vibrio, by a bacteria called Vibrio cholera. And this bacteria can secrete an exotoxin called the cholera toxin. Toxin means maddesam. cholera toxin, and cholera toxin causes cholera disease, مرض الكوليرا. We have also an example of an exotoxin. We have botulinum toxin. The botulinum toxin is released by a bacteria called Clostridium botulinum. Now, many other bacteria are also, this botulinum toxin, it's an exotoxin secreted by a bacteria called Clostridium. And this causes what we call, in Arabic, it's called تسمم منباري. Okay. This toxin causes a condition or a disease called botulism. In Arabic, تسمم منباري. Okay. We have also other, so this is produced by a bacteria called Clostridium. It's mentioned in your book, botulinum. Okay. An example of exotoxin, endotoxin, when the bacteria dies and its cell wall breaks. Gram-negative bacteria, if you remember, the cell wall of a gram-negative bacteria contains what we call lipo. So the outer, there's an outer membrane, and the outer leaflet of the outer membrane contains what we call a molecule or molecules called lipopolysaccharides, or LPS. So LPS is an example of an endotoxin. So this is an endotoxin. Okay. Examples of bacteria that causes that have endotoxins is the salmonella. Commonly found in, let's say, in shawarma sandwiches. Okay. So we have Salmonella typhi, can cause typhoid fever, or they can cause food poisoning. تسمم غذائي. Food poison. In Jordan, we've had many salmonella outbreaks or food poisoning outbreaks caused by this bacteria, especially in restaurants selling shawarma, okay, or chicken shawarma sandwiches. So this bacteria is found. It's very common in eggs or in chicken in general. Okay, and sometimes it might find its way into yes, raw chicken or raw eggs, because mayonnaise is made of eggs, and if the eggs are contaminated with this bacteria, the mayonnaise that is made from contaminated eggs might also become contaminated with this bacteria. Now, some bacteria, they become pathogenic because they have what we call virulence genes. Okay? So virulence genes, these are the genes that are, let's say, responsible for the pathogenicity of many, or pathogenicity of bacteria, pathogenic bacteria, let's say. So some bacteria, they can acquire these virulence genes, the genes that will make them pathogenic and cause disease in humans. So virulence is defined as the pathogen ability to cause disease to the host. And here, the pathogen we're talking about bacteria, bacterial pathogens. Bacterial pathogens. Okay. Some bacteria, they can acquire virulence genes by horizontal gene transfer. So sometimes this E. coli, remember when I told you that we have millions of prokaryotes inside our intestine? We have some of these prokaryotes is the E. coli. But this E. coli is basically not harmful. غير ممرضة. But some E. coli, they can acquire virulence genes by horizontal gene transfer, like transduction or transformation, and they could become harmful bacteria or E. coli that cause disease, okay? Or disease-causing E. coli. Disease-causing E. coli. So here we have horizontal gene transfer. So basically, we have virulence genes might transform a non-harmful or a safe bacteria or not harmful bacteria into pathogenic or disease-causing bacteria. So, for example, there's a type of E. coli called E. coli O157H7. This is a highly, it's a dangerous bacteria, dangerous type of E. coli. Okay, and it can cause bloody diarrhea. Ishal. Diarrhea means ishal. Bloody means there will be blood and diarrhea. Okay. So we have E. coli that is not harmful, does not cause a disease, but also we have E. coli types that might cause serious disease. So scientists, since they discovered prokaryotes in nature, they have been using some of these prokaryotes for research and for technology. So we, as humans, for example, have been using certain types of prokaryotes to make certain types of food. So, for example, many like we have been using bacteria to make yogurt. So yogurt اللي هو لبن, for example. So yogurt, for example, requires the presence of certain types of organisms in milk. Yogurt, cheese, okay. Let's say certain types of cheese, yogurt, pickles, مخللات, okay, and many other types of food, okay. But also we use, for example, E. coli for gene cloning. We have scientists are now using CRISPR-Cas system from bacteria for gene editing. Also, we can use bacteria, for example, to make useful products like natural plastics, or bacteria to clean the environment for us. So there are hundreds or thousands of applications where bacteria are used for useful purposes. Many bacteria are also being used for producing what we call biofuel. So biofuel is a fuel made from agricultural and waste biomass. So some types of bacteria, they can, let's say, they can convert the agricultural waste and certain types of plants into ethanol, and this ethanol is used as biofuel, okay, or as fuel or source of energy. Simple. Okay? Some prokaryotes are used in bioremediation. Bioremediation is basically the use of organisms to remove pollutants, ملوثات, from the environment. Okay? So this is an image of two workers spraying a lake shore contaminated with petroleum oil. They are spraying this petroleum oil contaminated shore with bacteria, because the bacteria, they can degrade petroleum oil and clean this environment. Inshallah, there's a course called biotechnology or basic biotechnology. I actually teach this course, basic biotechnology. In this course, technology, it's a BT 2030 course. And there's another course called microbial biotechnology. In these two courses, I think for 3-1, in these two courses, you will learn about the wide range of applications of prokaryotes, okay? The biotechnology applications of prokaryotes in industry, in medicine, in agriculture, in food, and many other applications. So the next chapter is about viruses. This chapter, inshallah, we're going to learn about the genetic and the phenotypic characteristics of viruses, the different types of viruses, the major types of viruses, and their life cycles. And at the end of the chapter, we're going to learn about some of the disease caused by certain types of viruses. So what's a virus? A virus is considered, it's not considered an, let's say, a living organism. It's considered an infectious particle. So we have genes surrounded by a protein coat. viruses, they do not carry out metabolism on their own. They need a host cell in order to carry out metabolism. They are much simpler in structure than prokaryotes. They are smaller than prokaryotes in general. They cannot reproduce or do metabolism or carry out metabolic reactions outside the host cell. So outside the cell, they are considered, non-living particles. But once they are inside the cell, they start reproducing and carry out chemical reactions. So they are considered live organisms once they are inside the host. So basically, you could say that viruses they have a life between life forms and chemicals. Okay, so they are living and non-living at the same time. So viruses were first directly discovered before even scientists could even see them or see them under the microscope, for example. And the discovery of viruses was done by a German scientist in 1983. And the first virus discovered was a virus causing disease to tobacco plants, نباتات التبغ. So there's a disease called tobacco mosaic disease caused by a virus called tobacco mosaic virus, or TMV. Now, this virus can infect tobacco plants, leading to a change in the color of their leaves. So their leaves will look like a mosaic, will have a mosaic coloration. So they have different, let's say, here, this is the mosaic variation. So the leaves, they have these yellow dots, but also they have these green dots. This is what is meant by mosaic. Mosaic فسيفساء. Okay? So Adolf Meyer, in the 1983, he was doing experiments on tobacco plants with this disease. And he showed that he could transmit this disease. So this is how the virus affects the plants. This is a normal tobacco plant, and this is a mosaic or tobacco mosaic or a plant that is affected by the virus. So Adolf Meyer, he showed that he could transmit the disease, the tobacco mosaic disease, from one plant into another. Okay, how? This is, for example, this is the experiment done by Adolf Meyer. So what he did, he took some of the leaves infected with the disease, okay, and he rubbed them with healthy plants, okay. So now the healthy plants will become infected with the virus. So basically, he took the virus from the leaves of infected plants, okay, and he rubbed them. So let's say this is not actually the experiment done by Adolf Meyer, but it's a similar experiment in principle. So Adolf Meyer was the first to show that he could transmit this disease, but he didn't know at that time that he was actually transferring viruses. So he took this plant, this leaf, for example, that is infected with the virus, and he rubbed it. Okay, so rub means he rubbed it into healthy leaves. Okay, so these are healthy. I don't know what's the meaning of rubbing Arabic. Rubbing in Arabic means farq. Okay, and this led to the transmission of the disease from the this plant into the healthy plant. So when he took this leaf that showed signs of tobacco mosaic disease and rubbed it against leaves of healthy plants, the leaves of healthy plants, they also became mosaic, and now they have the disease. Okay. Again, this is in 1983. Other scientists, of course, Adolf Meyer, he did not know that he was actually transmitting viruses from one infected leaf into the healthy leaves. Later on, another scientist, Martinus Bujernik, he did this experiment to show. He's a Dutch scientist. He did this experiment to show that the disease is caused by viruses but not bacteria, because after Adolf showed that he could transmit this disease from infected plants into healthy plants, other scientists, they suggested that it might be bacteria causing this disease. But Martinus did this experiment where he took infected leaves and prepared what we call a sap. So he extracted the liquids or the sap from the plant. Okay, so he you could say he blended or grinded the plant leaves to and prepared an extract or a liquid called the sap, and then he filtrated the sap through a filter. And this filter here, so here we have a filter. Okay, this filter contains microscopic pores, ثقوب مجهرية, microscopic pores. And these pores are small, very small. They cannot allow the bacteria to pass. So they filter out bacteria. So the bacteria is trapped on top of the filter. Okay? So the sap, basically, the liquid sap will be moving through the pores. But if there is bacteria in the sap, okay, the bacteria is bigger than the pores, and the bacteria will be trapped on the surface of the filter. Okay, so this is the filter. But if there are viruses, okay, viruses are very small. They are smaller than. So this is a virus, and the virus can go through these microscopic pores. So the viruses can, like the sap, like the liquid, they can go through these tiny pores or microscopic pores. So the idea is that here, by filtrating the sap, the bacteria will remain on the filter, while viruses will be found in the what we call filtered sap المنقى. Okay. Now, when the filtered sap was taken and added to healthy or rubbed into healthy tobacco plants, and the tobacco plants, they developed this disease. This suggested that this disease is not caused by the bacteria, because the bacteria are not found in the filtered sap. There's something else, which turned out to be the tobacco mosaic virus. Okay. So here we have the tobacco mosaic viruses, and this experiment showed that viruses, but not bacteria, are the causative agent of this disease in tobacco plants. Later on, in 1935, Wendell Stanley, another scientist, he was able to show that tobacco mosaic disease was caused by the tobacco mosaic virus. So he used, basically, he was able to show that the tobacco mosaic virus was the causative agent of this disease. So he was able to crystallize the viruses, and he was able to see the viruses under the electron microscope. Okay? So he converted the viruses into crystals that could be seen under the microscope. In terms of structure, viruses are not considered cells. Simply, viruses, they consist of nucleic acid. Some viruses, they have RNA. They are made of RNA. Some, they're made of DNA. So the genetic material of viruses might be RNA or DNA. Some viruses, they have single-stranded RNA or double-stranded RNA genetic material. Some viruses, they might have double-stranded DNA or single-stranded DNA as genetic. Whatever the case, the nucleic acid is protected by a protein coat. And in some viruses, there will be an additional layer made of membrane. So some viruses, they have protein coat and membranous envelope. In terms of the viral genomes, as I told you, some, they have double or single-stranded DNA. Some other viruses, they have double or single-stranded. So viruses are classified as DNA viruses or RNA viruses. And the genome could be single, linear, or circular. Some viruses, they have 3 genes, while others, they have thousands of genes or hundreds of genes in their genome. So the protein coat, or the proteins that surround and protect the viral genome, they form a structure called the capsid. The capsid is basically the protein code, and it is built from protein subunits called capsomeres. Now, different viruses, they have different shapes of these capsids. Okay. So some capsids are helical or icosahedral. So here, in this picture, we have different types of viruses. This is, for example, tobacco mosaic virus, which causes disease in plants. We have adenovirus and influenza viruses, they cause disease in humans. And we have bacteriophage, which causes disease to bacteria, which can infect bacteria. And as you can see, this is the capsid shown here in the purple color, and these are basically the capsomeres. This is another shape of the capsid. So here we have a helical capsid. Here we have an icosahedral capsid, as you can see, and these spheres, small spheres, are the capsomeres. Some capsids, they have also protruding proteins called spike proteins. Usually these spike proteins, they allow the virus to bind on the surface of the host cell. This is an example of an enveloped virus. So here we have the genetic material of the virus. As you can see, it consists of segments of DNA. And then we have this membrane envelope. And on the surface of the membrane envelope, we have these glycoproteins or spike proteins. Again, these spike proteins, they allow the virus to bind to human cells to cause influenza. And the influenza virus genome is segmented double-stranded RNA gene. And as you can see here, this is a virus or a bacteriophage or a phage that can infect bacteria, especially E. coli. So this one can infect E. coli. It looks like a spider with tail fibers like legs. They allow the attachment of this virus to the surface of E. coli. And then we have the tail sheath. This is like a syringe that will inject the DNA that is found in the head of the virus into the inside of the bacteria. Okay. So as you can see here, we have an example of 4 viruses with different, let's say, genetic material. The tobacco mosaic virus contains a helical single-stranded RNA genome. Here we have double-stranded DNA, double-stranded DNA. So here we have different shapes of the capsids. Okay, this one has an envelope. This one does not have an. Okay, this is a helical shape of the capsid. This is icosahedral shape. So let me stop here. Inshallah, we'll continue tomorrow.